Okay. I hope that you are all doing well. We are going to do an experiment in lab tomorrow, and there will be a lab handout posted before lab tomorrow, so just take a look for that. Today we are going to talk about the other side of peripheral tolerance, which is sort of how tolerance breaks, and how we can get to autoimmunity. So if we think back to last time, we talked about two different types of tolerance. We had both central tolerance and peripheral tolerance. And when we thought about peripheral tolerance, we talked about clonal deletion, clonal helplessness, clonal deviation, clonal suppression, clonal anergy, and clonal ignorance. So these were sort of all of the different mechanisms of tolerance that we saw on Monday. Autoimmunity, of course, results from some kind of failure in self-tolerance. And it could be failures related to any of the things listed here. Hold on. No. There. Okay. So as I said, the specifics of the tolerance failure really could be failures of any of the aspects of tolerance that we mentioned last time. And it's a pretty good list. There's a bunch of mechanisms of tolerance that we can come up with. What you can see is that we often see those failures either having something to do with a genetic factor or some kind of environmental factor, often a little bit of both. That will lead to that kind of failure of tolerance and autoimmunity. But we've got a bunch of tolerance mechanisms that we talked about last time. And that might make you think that, this should not happen so much. But some of these autoimmune diseases are decently common, perhaps more common than you'd think, given the number of tolerance mechanisms. Last time we said, oh, they're not very common if we think about peripheral tolerance, or central, just central. But now we added peripheral on top, and it's like, okay, but then now how do we fall through those cracks? To the point of 1 in 50 people having psoriasis, or 1 in 100 having rheumatoid arthritis. We can generally divide up some autoimmune diseases based on whether they seem to have involve issues with peripheral tolerance or issues with central tolerance. And so I'm going to talk today a little bit about first a couple of diseases that are usually thought of as defects in central tolerance. And then we're going to talk about some diseases that are defects in peripheral tolerance, or we're going to talk about some defects in peripheral tolerance. And then depending on time at the end, we're going to talk about some other issues of autoimmunity broadly and some individual autoimmune diseases that are just the ones most immunologists know some stuff about. There is one other point that I think is really important to make with a lot of these discussions of autoimmunity. And I struggled with where to put this point and how to put this point in this lecture. And so I ended up coming up with, I was just going to say it early. So I'm just going to say it early, which is that when we think about autoimmune diseases, there are two different parts of autoimmunity we can think about. We can think about what is the initiating event, or what is the initiating factor? Why is it that some cell slipped through the cracks? But the other thing that you should be aware of is that most patients with autoimmunity don't have one cell slip through the cracks and go to the doctor's office the next day. That one cell might slip through the cracks. That one cell might eventually get activated, proliferate, do some effector function, cause some damage to tissue. And only after there's been a certain amount of tissue damage will that patient actually be diagnosed and treated. And so when we think about autoimmune diseases, we can often look at a patient and say, wow, this patient seems to have some CD8 T cells causing them problems. This patient has some tissue damage that seems to have been caused by antibodies, or whatever. And so we can sort of say, in this particular case, we can say that these immune effector mechanisms are the problem. And at the end today, when I talk about some specific autoimmune diseases, I'm often going to be talking about this is the effector mechanism that causes the problem in this disease state. But we can also think back to why did that first cell break tolerance? And we can't do a great job in describing it to any one individual why exactly that tolerance break happened. We can talk about some general ways that we know tolerance breaks, but when you go to your doctor and get diagnosed with rheumatoid arthritis, was on the previous slide, your doctor can't be like, well, 7 years ago, there was a T cell that had this happen to it on a Tuesday. We can't identify. We can do some sort of statistics and other epidemiological types of things to say these are the things that are likely related. These things seem to be related. But we can't necessarily pinpoint in each individual case what the initiating event is. In the first part of this lecture, I'm going to be more talking about kind of some general ideas of initiating events. It's very rare that we can say this particular event happened in this particular patient. But these are sort of the theories, the big picture theories of what happens. And that's always a little bit uncomfortable for immunologists because patients will maybe look at some of this stuff and be like, well, what happened to me? Why did this happen? One of my friends has a daughter with diabetes, with type 1 diabetes, which is a very famous autoimmune disease. And my friend pulled me aside and was like, well, what did I do wrong so that my daughter had diabetes? There's sort of this question of what is the initiating event and how could you have made it go away? And we can't know in most cases. And as I think you will see in some cases, the answer is really bad luck, at least partially. And so What I want you to just realize is that at the beginning here, we're going to talk about some kind of big picture ideas about initiating events, but how we tie one of those initiating events specifically to any individual patient's case is not really something we can do, or something we can only do in extremely rare circumstances. So in that sense, realize that the two parts of what we're going to talk about today, the initiating events and the effector mechanisms in specific diseases, yeah, they're tied together theoretically, but we can't make a direct linkage with all of them. The first part of this is that I want to talk about what happens when individuals have defects in central tolerance. If someone has a defect in central tolerance, what you might imagine is that they're probably not going to have one T cell be unlucky and fall through the cracks. They're probably going to have many autoreactive T cells that are not dealt with because of this loss of central tolerance. Those many T cells, or many B cells, again, here you can see my past life as a T cell biologist come through. Those many T cells are probably not all going to attack the same self-antigen. They're probably going to attack a whole bunch of different self-antigens. And as a result, that patient is probably not going to have one type of autoimmune tissue damage. Instead, that patient is probably going to have autoimmune diseases that attack multiple organs. They might have multiple autoimmune diseases, not just one, if they are having an issue with central tolerance. There are two particularly well-characterized autoimmune diseases that involve an issue with central tolerance, and thus the patients have a whole bunch of different symptoms of autoimmunity. These, I guess, are unique in that, yeah, we can actually tell in most cases, in both cases, we know the patient is missing this gene. So I guess in that case, we do know kind of the initiating event, or at least part of it, is that we know the patient was born without this one gene. So one of these diseases is a disease called APEX. You can see APEX in the upper right corner. You do not need to know what APEX stands for, but I will tell you that it stands for autoimmune polyendocrinopathy with candidiasis and ectodermal dystrophy. That name, that big long name that I just said, puts together a bunch of different symptoms. And that's because these patients have this large number of different symptoms because it's we see a defect in central tolerance. Specifically, these patients are missing air. So they don't have the gene encoding air, or they have a mutation in that gene, so they can't make the functional protein. And as a result, they cannot make all of the transcripts that are needed in the thymus. So there are some antigens that are not present in the thymus for negative selection. Those T cells never get negatively selected. They make it to the periphery. They lead to autoimmunity. When we look at these patients, each patient seems to have a different pattern of symptoms based on sort of which T cells made it out and got activated. So that's going to be a little different for each patient. So often there are some issues with the endocrine glands. The PE is polyendocrinopathy. So you can see that many patients will have hypoparathyroidism, many will have adrenal failure, many of the women will have ovarian failure, and so you can see sort of there are going to be these distinct different symptoms. Also, 100% of the patients have candidiasis, that's the C in APEX, which is recurrent yeast infections, particularly in the mouth. A lot of them have weird findings in their fingernails, which you can see here, as well as some weird things with teeth and some other sort of physiologic findings. You can imagine this might be a little bit tricky to diagnose, as the patient is having all of these really diverse symptoms that they may or may not immediately realize are related to one another. The other big difference that we can, or the big defect in central tolerance that we see is in another disease that's known as IPEX, and you can see IPEX listed there. Oops, oh. I didn't know that happened. I have now learned a new PowerPoint feature. For our Zoom classmates, if you put in the chat what seems to be wrong about screen sharing, that would help me diagnose. And for your future reference, if you're ever giving a PowerPoint presentation and you type the letter W, you get a white screen or a new slide, because that's what I just did. So now we all know that. In any case, this disease, also a defect in central tolerance, is known as IPEX. IPEX is found mostly in boys because this gene is X-linked. And IPEX actually stands for immune dysregulation, polyendocrinopathy, enteropathy, X-linked. No polyendocrinopathy is in there again. It seems like from these kinds of diseases, we realize the endocrine glands seem to be really sensitive to some of this destruction. In any case, IPEX is caused by mutations in FOXP3. And so these patients, mostly boys, do not make regulatory T cells. And so they are both unable to make those regulatory T cells in the thymus or in the periphery. These patients, as it says on the bottom here, often have autoimmune enteropathy, so GI tract issues, eczema or other dermatitis, nail dystrophy, so again, weird stuff with the fingernails, autoimmune endocrinopathies, so endocrine gland things, autoimmune skin conditions like alopecia or pemphigoid, severe enlargement of secondary lymphoid organs, insulin-dependent diabetes, food allergies, and infections. Most affected children die within the first 2 years of life. And so this is a very severe syndrome where we are seeing this loss of regulatory T cells, which leads to a loss of central tolerance and sort of this tolerance across the board. All right, quick troubleshooting detail. All right, stop share. Share screen. Oh, wait. All right, did that fix everything? While we see if things have been fixed, I'm going to tell you that alternatively, we can see situations where peripheral tolerance is the thing that goes awry instead of where central tolerance is the thing that goes awry. These often have more impacts on what we see with kind of a lot of our individual patients who don't have sort of genetic issue. These also, as I said, oftentimes have a certain element of bad luck involved in them of you had to have a particular T cell get through a particular peripheral tolerance mechanism, and that T cell also had to come in contact with its antigen in one of these situations that we're going to see. And so, you know, sadly, that happens to a fair number of people. In many cases, we see situations where we think, or we can sort of make some hypotheses that those effects have to do with some infection that someone may have had many years before. So what may have happened is that there may have been a patient who had an infection that led to one of these peripheral tolerance breaks. And that also, during the time of that infection, the patient had the wrong T cell interact with the wrong self-antigen and get activated. And so in almost all of the cases that I'm going to tell you here, we might imagine ways that infections could be those initiating events. All we can really do is, in some cases, we can say, well, it turns out that in many people who have reactive arthritis, we can find evidence that sometime in their past they were infected with one of these microbes. But we can't actually say for definite sure that, oh yeah, it was that time 8.3 years ago where you were infected with salmonella. You know, when you go in for to get diagnosed with reactive arthritis, your doctor's first question is, tell me about stomach aches you had many years ago. That you're not going to make those links. And so, again, sometimes we can say epidemiologically there is more evidence in the autoimmune patients than in not autoimmune patients of some of these infections, but it's a little bit tenuous. But you will see that in many of my cases, an infection is one of the things that seems to be an initiating event for breaking the mechanism of peripheral tolerance. And so I'm going to do this sort of thinking through some of our different types of peripheral tolerance that we thought about last time. We talked about 6 mechanisms of peripheral tolerance last time. 2 of them are not really going to show up much today. It's a little harder to break 2 of them. The 2 that are sort of harder to break that we're not really going to see show up today are clonal deletion and clonal helplessness. Because in both cases, breaking those would involve like bringing a cell back from the dead, right? Like the cell got deleted in clonal deletion, or the T cell got deleted so it can't provide help in helplessness. In both of those cases, you can't undelete the cell. So it's sort of hard to come up with sort of clearer examples of how those break. The other 4, we're going to see examples of how they break. I will also put in that I'm going to next talk about clonal deviation, or a story about kind of clonal deviation and what we think may happen in a particular disease state to lead to that clonal deviation break. I will also say, so I want you to remember that when we think about clonal deviation, it's really about shifting T cell subsets. So maybe in a healthy situation, you make a Th2 response, but for some reason, you have made a pathologic Th1 response instead, when you should have made the healthy Th2 response to self-antigen. So it's about shifting subsets of helper T cells. Here, I'll sort of be lumping deviation and suppression, the 2 of our 4 mechanisms, together. Because suppression is also can be like, oh, you were supposed to make Tregs, but you didn't. So those 2 can sort of be very similar in that they're both about messing up which kind of helper T cell subset you have. This particular disease is, or the disease states that I'm going to tell you about are also a little bit getting an asterisk by them. Technically, a autoimmune disease is one where you are responding to a self-antigen. And there are sort of more allergic type diseases where you are responding to something foreign but that you shouldn't respond to, like me and pollen. I'm not a tree, but pollen's not trying to hurt me. And the diseases I'm going to tell you about in this case are actually both kind of weird borderline diseases. But they're also the ones where we actually know some things about deviation. So I'm going with them, even though if I was going to be super picky, I would be like, do I call those an autoimmune disease? And have like a philosophical conversation, whatever. And so you can see one sort of example of this here, and I'm going to talk about a similar example where things are even more worked out on the next couple of slides. But this is particularly looking at some Crohn's disease cases, where what was noticed is individuals had to have a particular gene mutation in this gene, ATG16L1, and had to get infected with a particular virus. And so what you can see is that there are 4 different situations with these mice that these investigators were looking at. Some mice were wild-type mice. They had normal genes, and they got infected with this virus. And they could actually be infected with viruses of 2 particular variants. So slightly genetically different versions, same virus, just 2 variants. And those mice, had some acute disease from the virus, and then they got better, and they're fine. You can also imagine mice that had genetic susceptibility. If those mice got infected with one variant of the virus, they were fine. But if the mice got infected with a different variant of the virus, they seemed to get Crohn's disease. And so this is what I wanted to say about the idea that it's a little bit hard to kind of come up with the initiating event. Was it being, can I say you have to have, this gene gives you Crohn's disease? Well, if you don't have the gene, yeah, I guess. But you can also not have Crohn's disease if you had that gene. You had to have this, be unlucky enough to have this gene, and be unlucky enough to get this variant of this virus, to have this sort of tolerance break. And so this is sort of the idea of how this is going to be a bit multifactorial. And the idea is that we're sort of seeing multiple things coming together. And we've begun to understand this actually even better in a model of celiac disease. And so what this group has shown, they have some follow-up papers since this one, but this one gives us the idea. With celiac disease, and celiac disease, to sort of simplify for our purposes right now, involves you having a dietary antigen that you eat, and eventually that leading to an immune response to a particular antigen. There's some chemistry that happens between the eating and the antigen, but we'll say right now there's a dietary antigen. Many of us eat that dietary antigen. Most of us, pretty much all of us, eat that dietary antigen. And we are okay. In fact, what usually happens is we are exposed to the dietary antigen, And just like we saw with our general mucosal immune response, any antigen-presenting cells in that area might make more of a tolerance type of response. We're going to get sort of dendritic cells maybe that are going to make more immunoregulatory cytokines. And so any T cells that get turned on in response to that dietary antigen might be Tregs. This is the general idea of oral tolerance that we talked about before, that typically most of our dietary antigens, we are going to make this oral tolerance Treg type response to. We aren't making responses to most of the dietary antigens that we encounter. In fact, if you go back and look at the slides I showed you when we talked about mucosal immunity and oral tolerance, I actually showed you this slide with just that part, with the rest of this with a box over it. Because that's what it should look like, and that's sort of what normally happens. But in some people, they have an unlucky event happen. Perhaps they have never really turned on T cells to this antigen before. Maybe they have not really encountered this antigen much before. So they don't already have a whole bunch of Tregs in response to this antigen. So you might imagine, a kid and the first time they eat the antigen. Obviously, we can't actually go back in time and be like, it was that meal. But we'll imagine in our heads that, we've got this kid and it's the first time they ever have this dietary antigen. So it should have been the time in their life where they made a nice, happy Treg response. At that particular day, that kid might have had a GI virus. In particular, where this has been shown really well, is this virus called reovirus. When that happens, not only are we going to be making some immune responses to the dietary antigen, we're also going to be making some immune responses to the viral antigens. We're going to have a bunch of different innate immune cells, like dendritic cells, here's one here and here, that are going to get activated and be doing their thing in this location. Because, oh my gosh, we have this diet antigen and we have this virus antigen. We have a lot of things to do. Both of those innate immune cells are going to make cytokines. This one might make some regulatory cytokines. This one might make some inflammatory cytokines. And now, when this T cell that sees the dietary antigen comes to this location, it's not just going to get Treg cytokines and say, become a Treg. It's going to get inflammatory cytokines. Because there's inflammation in this area. And the cytokines are not necessarily going just from one cell to exactly one other cell. It's sort of going to be an environment where there's a bath of cytokines around. The T cells are going to sort of go into that bath. They should have become a Treg. But because they happened to see their antigen for the first time on a day when these other cytokines were around, those T cells did not become Tregs. Instead, they became Th1 cells. And then the next time this person eats that dietary antigen, we don't have Tregs around to turn off responses. We have Th1 cells around. And those Th1 cells are going to come in and they're going to start damaging the GI tract. All because the wrong cytokines were there on that wrong day. And so what I hope you see here is the role for sort of bad luck. Of you had the wrong particular virus on that day where you first saw that antigen. Right? And it's kind of hard to go back when you actually show up at your doctor's office many years later when you've had a lot of damage to your intestine. Because you're not going to show up to your doctor's office and be like, Doc, I made a bad T cell. You're going to show up when you have symptoms. And those symptoms are going to happen when the T cells have done a lot of damage. It's going to be really hard for the doctor to be like, well, can you please tell me about the first time you ate this food, and did you also have a stomachache that day? Right? Like that's not how this is going to work. But really, that's sort of bad luck is likely what happened in this case. You can see that sort of thing here as well, where we might have our dietary antigen, where we should be making a Treg response, but because we have the dietary antigen and the virus at the same time, we might have some innate immune cell. We could even have the same dendritic cell that's phagocytosing stuff, phagocytose both of them. So that dendritic cell could be actually making our inflammatory cytokine and showing that antigen, and we can get suddenly an immune response that we should not have gotten against this antigen. Remember, this is showing this as a Th1 response. Remember in your table of helper T cell subtypes and responses that to get Tregs, you needed TGF beta, and to get Th17s, it was TGF beta plus inflammation. It was TGF beta with some inflammation on the side versus TGF beta alone. So you can kind of see how you add the inflammation on the side, suddenly you've switched the type of T cell response that you might be getting. And so there is a decent amount of evidence that this is largely something that is involved in celiac disease. And so you can see, again, if you're the physician and you have this patient and you're like, why did this happen to me? Kind of hard to actually be able to like explain it to the patient other than bad luck happened to you, right? Like if you really look about these events. I will also point out, it's not really shown here as well, but typically in these cases, we also tend to see that the patients have a particular type of MHC as well. So it's not, this isn't in everybody that encounters that antigen and that virus for the first time on the same day. It's only the people who also have a particular type of MHC that can present this dietary antigen in a particular way. So in pretty much every case I'm going to tell you about, there is also an MHC component where the self-antigen in question has to be promoted or presented by a particular MHC. So everybody doesn't have the MHC to present this antigen, only some people do. So it's actually, you had the bad luck of having the wrong MHC and eating the wrong antigen on the day you had the wrong virus, all put together. So you can also imagine that sort of tracking this or predicting this patient is going to have this disease can be really hard. We're going to mostly track those patients at the end after they've had a lot of damage, which might be harder to fix than if we could actually find them on the day that they made the bad T cell. So some of the most sort of frequently discussed aspects of tolerance breaks and ways that we think about tolerance breaks have to do with clonal anergy and the ways that we might have clonal anergy go awry. There are two general ways we can imagine clonal anergy going wrong. So I'm going to tell you two different. Wow, Siri did a PubMed search. That was nice of her. I'll tell you two kind of different ways that clonal anergy can get messed up. So you don't necessarily have to try to like make them go together. They're just two different ways anergy can get messed up. And so remember with anergy that we have to have our T cell get signal 1 and signal 2. If our T cell gets signal 1 alone, it will be anergized. And that's what should happen in a self-reactive case, particularly because there's no pathogen around to allow for the production of B7 to give that signal 2. one mechanism of breaking anergy is described here. So we can imagine, again, a situation where our cell is presenting some self-antigens. Remember, especially on MHC class 1, we are presenting on self-antigens. We are presenting self-antigens 24/7. The cells never are missing MHC class 1. That was one of the multiple choice questions on the most recent exam, and I put it on there because that point that we are always presenting is so important, both in terms of NK cells and in terms of autoimmunity. Always presenting. You're never. If you actually had cells that were ever missing MHC, that would be really bad. So we're always presenting self-antigens, but normally we should have this situation where we're presenting some self-antigens, but we have no infection. We have no PRR stimulation, so we have no B7 on the surface of this cell. We might stimulate the T cell receptor and give signal 1 to this T cell, but we don't have anything for giving signal 2, and so we're getting anergy. Now let's imagine a situation where our antigen-presenting cell does see a microbe, or where there is a PRR on our cell, or the PRR getting triggered in our cell. Now our cell is going to make B7, right? Our cell doesn't make one copy of the B7 protein that is somewhere on the surface of that sphere. So imagine the cell as a sphere. It doesn't just have one B7 somewhere in like that crowded mix of proteins. It makes a bunch of B7s, right? And B7, like any transmembrane protein, diffuses freely in the membrane. Every protein diffuses in the membrane. So it's just sort of traveling around that sphere in different ways. Our cell also doesn't have one copy of the MHC molecule on its surface. It's got a bunch. We know that both from it's got all of moms and all of dads, but even those, it has more than one copy of each one. It's got a bunch of copies. But we'll imagine in our head it's 100. I don't know what the number is, but we'll imagine it's 100. So we've got 100 MHCs on the surface presenting peptides. And it's whatever peptide that MHC got in the ER, if we're talking about class 1. So if we think about a case where we have an infection, we got those 100 MHCs on the surface of the cell. What if, would you believe me if I told you that 97 of those 100 had peptides from the virus? If anything, that you might be like, that's kind of a little high, because there's a lot of peptides in that cell. Like, there are still going to be some peptides made from self-proteins. Some of them are going to randomly end up on the MHCs. So maybe it's only like 50-50. So here you can see 50-50. We're presenting some self, the yellow from before, and some micropeptides, the beige, or the brown, because it came from this brown microbe. So there's a micropeptide, there's the self-peptide, the same self-peptide as before. You're not going to get like 100 and 0. There's still going to be some self-peptides presented by that cell, because it's presenting many MHCs at once. And those MHCs are diffusing around the membrane, as proteins do. Does the B7 know, oh, I'm only allowed to go with this MHC plus peptide. I'm not allowed to go with this MHC plus peptide? No, the B7 doesn't know that. The B7 is just diffusing around the membrane, as is the T cell. And so this cell that starts making B7 can turn on a T cell that responds to the microbe, but it could also turn on a self-reactive T cell with one of those self-peptides that it's presenting and its B7 that it's seen. Everybody always wants a name for this one, and there is no like specific name. It's like, I don't know, wrong B7 sharing or something. The idea is just that the cell is making a bunch of B7 because it did get infected, but it uses that B7 to co-stimulate a self-reactive T cell for one of the self-antigens it's presenting instead of one of the foreign antigens. Again, you can imagine there's a little bit of bad luck going on here. So yeah, this antigen-presenting cell had to present a self-peptide along with B7. And let's pretend, again, in our head, that that happened for one day, right? For one day, this presentation was happening. During that one day, this cell has to come across the matching T cell. Because sometimes people look at this and they're like, oh my gosh, we should all be dead. This should, but no, there's a certain amount of, okay, so this cell had to be presenting that peptide during the brief period of time it was making B7, and during that brief period of time, it also had to run into the T cell that was going to recognize this weird this situation. Yeah?
So this just happens by chance?
Totally by chance. So again, now you're imagining as the physician, yes, 8 years ago, it just so happened that one of your dendritic cells made B7 when it shouldn't, and shared it with the wrong MHC. Like, you can't tell someone that. So people will be like, well, why did this happen to me? And the answer in some ways is like, bad luck. Right? Did you have a question, Jay? Okay. For some reason I thought I saw your hand, but I can totally make things up. So before I go on to the next way of clonal anergy going awry, I do want to point one other thing out. This is 100%, 99.999% conjecture on my part. There's a little bit of data that makes me think this, but no one has actually made the tie that I'm now going to make. But I want you to think about this tie, even if we don't officially have all the evidence for it. In many patients right now who have previously had SARS-CoV-2 or COVID, we are starting to see weird symptoms, in many cases that are called long COVID. In many past SARS-CoV-2 patients, we are seeing huge numbers of autoantibodies. Huge numbers of autoantibodies. And there's some suggestion that long COVID may be some emerging autoimmune symptoms in some patients, possibly from those autoantibodies. You might imagine that when you are making, as we've talked about, the SARS-CoV-2 immune response, where the immune system goes like out of control, way too high. We talked about the out of control innate immune response with SARS-CoV-2. You could imagine situations like what you're seeing here on the bottom and some tolerance breaks during that infection. You might also even imagine some cytokines that are being made during that crazy immune response that could be otherwise messing up tolerance. So I think that one could hypothesize that some of what's going on with long COVID may be some of these sorts of tolerance breaks because of the intense innate immune responses that we're seeing. This is, like I said, the autoimmunity data and some of the autoimmunity long COVID data is real in terms of it looking like autoimmune diseases, and there absolutely are a lot of autoantibodies in COVID patients, but exactly how I'm tying it here to tolerance breaks is totally hypothetical. Yeah?
I was just also going to say hypothetically kind of that since there's so many cytokines in the response that it could turn back on.
It also could totally turn back on an anergic T cell when you are making just so many cytokines, absolutely. And so sometimes when I have looked at some of the long COVID literature, I have thought about some of these exact tolerance break mechanisms and said, I wonder if that's what's going on. But you can, again, imagine it would be really hard to actually prove that in any individual patient. Go back in your time machine and in your magic school bus so you can watch the T cell and find the T cell that gets inappropriately activated. We're not going to do that. So you can, again, it's very challenging, but one could imagine some of these things being linked. The other way that we often think about breaking anergy is with something known as molecular mimicry. Whenever people think about ways that we might initiate autoimmune responses, they often go with molecular mimicry. They're always like, oh, I bet it's a molecular mimicry case. So I put molecular mimicry later in these slides because it's not always molecular mimicry, even if it's the one that people come up with pretty frequently. And what you can see here is a case where we have MHC presenting some peptide from a pathogen. Remember that that peptide is maybe 8 to 10 amino acids in length if we're talking about class 1, up to 20 if we're talking about class 2. Some of our group, some of the amino acids are pointing up towards the T cell. Some of them are pointing down in making contacts with the MHC. So in fact, the T cell maybe sees like half of those amino acids, like 4 in the 8 to 10, 9 in the 18. Not that many, right? Sometimes there are pathogen peptides and self-peptides that are somewhat similar. They may actually, when you actually get down to thinking about 4 amino acids, actually be pretty similar. So here you can see a pathogen peptide and a self-peptide that has part that looks just like the pathogen peptide. Maybe it's the same 4 amino acids. If the first time the T cell came in contact with this this MHC plus peptide, it happened to be this self one, there would be no PRRs, no B7. We'd get tolerance. Hooray. But if the first time the T cell sees antigen, it sees this one from the pathogen, the T cell is going to get turned on. That's what should happen. We get our T cell getting turned on. There is, in fact, signal 1 and signal 2, though that signal 2 is not shown here. Now this T cell is turned on. It's no longer a naive T cell. It is now an effector T cell. And that effector T cell is going to leave the lymph node, and it's going to go around the body, and it's going to look for its antigen being presented on MHC. And importantly, when we think about effector T cells don't need signal 2 anymore. Well, remember we talked about memory and effector T cells and how they're easier to activate. They don't require signal 2 for activation. So now that effector T cell is going around the body, it might see a whole bunch of cells presenting the pathogen epitope and be like, yeah, I'm going to kill them, and kill all those cells. But it also might see some self cells presenting this very similar antigen. And yeah, there's no signal 2, but we don't need signal 2 anymore. And so now that effector cell is going to kill the self cell and start to cause autoimmunity. And so this is sort of a fancy way of kind of thinking about cross-reactivity. And the idea of certain pathogen peptides looking similar to certain self-peptides. Again, you can imagine this is a little bit of a bad luck timing thing. If the T cell had seen the self thing first, we might have gotten tolerance. But because, as luck would have it, this T cell came out of the thymus on Tuesday, because remember, you make T cells throughout your life. Your thymus is making new T cells and new B cells throughout your life. Today, you just right now finished making a new T cell. I don't know what it is, but you just made one. Good job. If that T cell, the first time it sees its antigen is it sees the self version and doesn't get signal 2, it's going to be off. But if that T cell is unlucky enough that it hasn't gotten turned off, it hasn't gotten energized, and happens to see this first, that T cell is going to get turned on. And that T cell is going to cause autoimmunity. So again, it's a like, I'm sorry, did you have the bad luck of creating this T cell on Tuesday when you were 17 years old and then getting this infection on Wednesday? Like, again, we can't actually figure that out, but that's the kind of thing that might have to happen. We can actually see this in a bunch of different cases. So here you can see this idea, and this is actually very much something that we think is really the case, that in childhood one might have some kind of viral infection with the upper respiratory tract, and we're going to make a bunch of different T cells, both helper and CTLs. We're going to make a bunch of different antibodies. Those T cells and antibodies are probably all going to recognize different epitopes from that virus. So we're going to have many different T cells responding to many different pieces of the virus, many different B cells responding to different pieces of the virus. And maybe one of those T cells, 1 out of 100, has the bad luck that it also recognizes a peptide that is presented by the pancreatic beta cell. Then those T cells are going to go into the pancreas. We're not going to need signal 2 anymore, and we're going to have those T cells come in, and we're going to have those T cells start to destroy the beta cells in the pancreas. So they're supposed to be red, and they get black when they get killed. That's what this figure is showing. So you can see we killed all the cells in the one on the right. And now, if we have no pancreatic beta cells, we have a disease called type 1 diabetes. This is what type 1 diabetes is. And so this is sort of how we think this might work out. What you might also realize in looking at this is that in order for this to happen, you have to have that 1 out of 100 of your T cells that happens to cross-react with a peptide presented by the beta cell. That's probably not something that's going to happen in everyone. That's probably only going to happen in people with certain MHC types that can present a particular peptide. So you sort of have to have the unluckiness of having the wrong MHC type so that you can present the cross-reactive antigen and getting this particular virus. So maybe you have that MHC type, but you never get that virus in your life, so you're fine. Maybe you get that virus in your life, but you don't have the MHC type, so you never have response to this cross-reactive epitope. So you can see kind of multiple things have to happen to make this work. Yep?
Sorry, I just need to clarify. So there would be unlucky to have that a certain MHC type?
Yeah. So
what would be the MHC type that would be unlucky to have?
so let me see if I can draw this. My drawing might be terrible, but I'm going to try something. Okay, so here, there's a purple one here. There's a blue one over here. This is kind of a different green. Okay. There's my virus. It's made of 6 different peptides. Okay? Six different people from this class each have MHC class 1 molecules. I mean, everyone in the class has MHC class 1 molecules, but we're imagining 6 of them right now. And I can't draw, so those are my 6 MHC molecules, okay? Everybody has their own MHC molecule. So we each have a different one. This person, MHC, presents the blue peptide. from this virus because the blue peptide matches that cleft really well. And they don't present purple or green or the other ones because their MHC cleft doesn't match that. Do you get that? Okay, this person presents purple. This person presents green. This person presents pink. This person presents white. This person presents this green. So each person, because they have a different MHC type, tend to present a different peptide from the virus. Here's the pancreatic beta cell presenting its self-peptides. This pancreatic beta cell presents a white peptide. See, the cell's white. I drew it in white chalk. The person who made the response to the blue peptide when they got the virus, this T cell that responded to this blue peptide is not also going to respond to the beta cell. So that person's safe. This person made a response to purple. The T cell is not going to get confused between this purple and this white. This person's safe. This person, their T cell, they made a really good T cell response against green. The T cells that respond to green are not getting confused. That person's safe. But you can see there's one person whose T cells happen to make a response to a peptide that looks a lot like this peptide. And so that one person is now sort of at risk. If that person had never gotten infected with this, so you could say, well, part of getting diabetes is having this MHC, right? If you had a different, the MHC that bound blue, you'd be safe. But it's not just that you had to have this MHC, you also had to get this virus sometime in your life to turn on the T cells. So it's sort of this multiple things are happening. Does that make sense?
Yes.
Okay. We have also actually been able to map some aspects of this. We can see MHC with peptide, and we can look at how T cells will respond to MHC plus peptide. And so here you can see an alpha chain and a beta chain responding to, from the T cell, responding to MHC plus peptide. You can see kind of this little angle of the T cell, the angle of the MHC. Sometimes what we've noticed, and these are two different cases of T cells that are found in multiple sclerosis patients looking at MHC plus peptide, the T cell, if it will come towards the antigen presenting cell at the wrong angle, and we'll be able to actually think this looks like this foreign antigen if it's coming at this weird angle or this weird angle instead of this angle. So this is actually some structural things that we have seen in terms of cross-reactivity. What I'm showing you here about molecular mimicry has been about T cells and molecular mimicry, because I like to think about T cells. However, it is definitely not just something that happens with T cells. There are, in fact, situations where we can see antibodies that cross-react as well. There is actually a very famous case of this, where there are sometimes antibodies that are made to a particular type of bacteria, a Streptococcus species, and those antibodies can also bind to some structures in the heart and lead to destruction of heart tissue in a disease called rheumatic fever. This is actually, this one, sometimes we can actually really find the antigens and the antibodies and sort of see what the cross-reactivity is. So there are some well-documented cases on this, rheumatic fever being one of them. On Friday, I will have a bit more to say about autoimmunity. I'm going to pick and choose among the remaining slides, and then I will also move into talking about allergies. And it's always fun. When I used to teach this class in the spring, a lot of times, since it was the end of semester, it would be like April, I could give an in-class demo of allergies while I was lecturing. But less of a problem in November than in April. So I'll see you guys in lab tomorrow. There will be a handout posted before lab.